Sorry for the problem here. Thank you. And I also start to thank, I'd like to start by thanking Stefan Sarantis and all acting Motif for putting together this really exciting event. So my lab, so Sharon mentioned, is based at the Francis Crick Institute in London, and our main interest is to study epigenetic mechanisms which are important for cancer development. And we have a particular focus on chromatin. So we are now, I think, at a really great time to study how epigenetics contribute to cancer development. And this because over the past few years, a large number of studies, omics type of studies, have carefully profiled the different types of alterations that impact the epigenome in patients at various levels. And so we have all now this rich catalogs, and we can use them to go back to the biology. And this is basically what we do in the lab. So we try to understand which one among all those alterations are truly important for the disease. And more importantly, we try to understand how they really impact cell function, so how they translate into malignant phenotypes. So if we think about the evidence that suggests a functional role for epigenetics in cancer, there are mainly three aspects that support this notion, and all of them come with interesting questions. So the first one is that mutations in epigenetic regulators are frequently observed in patients, which suggests that they provide a selective advantage to the affected cells. Now the functional impact of this mutation is not entirely clear at present, but for sure we know that this, as a group, are among the most mutated genes in cancer. The second, and probably most direct evidence, is that in many cases, cancer cells become dependent on epigenetic regulators to sustain their malignant phenotypes. And over the past few years, a number of epigenetic vulnerabilities have been identified, and they're currently being explored for therapeutic purposes. And finally, another layer where epigenetic mechanisms play a functional role in the disease is in the regulation of differentiation states within individual cancers, which impact disease maintenance and progression. So today I'm going to tell you two stories that fit into this framework. The first one is published and focuses on the third theme here, whereas the second one is a more recent one and instead addresses the issue of mutations in the epigenetic regulatory network. So it is well established now that within individual cancers, only a subset of cells are actually truly mortal and can proliferate indefinitely, where other cells instead lose this capacity progressively through a differentiation process. And this is nicely visualized in this image I'm showing here. So this is from a study from the Broad Institute. This focuses on glioma, but very similar patterns have been seen also in other cancer types. This is a single cell transcriptomic analysis, and we can see that there is a subpopulation of cells here at the top of the hierarchy, and this is dividing cells and possess an undifferentiated phenotype. However, there are other cells also, they're less proliferative, and this kind of resemble the cells that we find in normal brain. Now, the differentiation process that occurs during tumor growth is obviously just a disorganized version of what happens in normal tissues. But the end result is the same, and it basically deprives cells of unlimited proliferative capacity. So starting from the notion that within individual cancer, we find cells in distinct differentiation states, kind of a basic question that we asked a few years ago was, well, how are the states defined at the epigenetic level? And more importantly, what defines whether a cell will keep dividing or eventually differentiate? This brought us to identify an integral component of chromatin, the linker histone H10, as an important regulator of tumor organization and maintenance in numerous cancer types. So H10 is a variant of the linker histone, H1, which is the neglected histone. So not many people actually think about that. So H1 is not part of the nucleosome, just sits on top of it, is as abundant as the core histones, but it's remarkably poorly studied. And this mainly because, well, that's actually been difficult to understand what it really does in the cell. So H10 is a variant. It's one of 11 different variants encoded by different genes. And what's special about this variant is that it's a replication-independent subtype, so it's transcribed throughout the cell cycle. And this is the one that accumulates in adult tissues. Now, what you find about H1 in cancer is you will see, I think it's a really nice example of the tight interplay that exists in the disease between genetics, epigenetics, and cell-to-cell signaling. And that really shows that what we see in an established tumor is it's really a combination and kind of the overlay of all this. So what we find basically is that H10, as I mentioned, is highly expressed in most adult tissues. You can see here a few examples. But activations of oncogenes triggers a strong downregulation of this protein. And although this is essentially basically a consequence of driver mutations and the transformation process, functionally this is very important because in the absence of H10, cells acquire a chromatin state that supports or favors the repression of a large number of genes, genome-wide, that collectively support and control self-renewal. This is a downstream effector of epigenetic signaling pathways, actually the same that leads to the silencing of H10, and also stem cell-related genes, such as, for instance, Wnt targets. And we've shown that ultimately this repression is mediated by changes in nucleosome occupancy at the promoter of these genes, which is induced and triggered by the loss of H10. And so in this state, and together with everything else mutations do, cells start to divide, keep dividing, and see the growing tumor. However, what we've also found is that these epigenetic states, which are established by the initiating mutations, are not stable. And during tumor growth, as a result of interaction with the tumor microenvironment, many cells re-express H10, and all this is reversed. So we are now signaling affecting epigenetics. And interestingly, this basically counteracts what the initiating mutations have done. So the end result is that if we look within tumors, we can find highly heterogeneous levels of the protein. Some cells express high levels, some low levels. And importantly, we see that then this affects cell behavior. We see that cells that are able to stably repress H10 during tumor growth are able to keep expressing the self-renewal genes and keep dividing. Whereas in cells where the protein is re-expressed, the self-renewal genes go back to being repressed, and this pushes cells towards differentiation. Now, conceptually, I think one interesting point that comes out from this finding is that if we think about the cells here, which are the differentiated cells within the tumors, well, these are obviously still mutated, right? So they still have the driver mutations that trigger all this in the first place. But if a repressive chromatin state is reestablished at the downstream effector of the oncogenic signaling pathway, then essentially mutations cannot exert their effect anymore. So this shows basically that epigenetic mechanisms that act during tumor growth can override gene expression programs that have been established by the initiating mutations. So with this in mind, we thought that maybe we could try to take advantage of this to essentially push this naturally occurring differentiation process to the extreme and make all cells differentiate. So we searched for compounds that would allow us to restore normal H10 levels within tumors. We did this by screening about 4,000 compounds using 2 different cell lines. And we did find a particularly good one, Xenostat. You can see it's very effective at inducing homogeneously high levels of H10 within tumors. And we could see activity of this drug across many different cancer types. Now, interestingly, Xenostat is an epigenetic compound. It's a HDAC inhibitor. It's not the only one that we found in our screen, which obviously gives us confidence in the results. But this is the one that we decided to focus on because this is a second-generation inhibitor. It's very potent, very stable, and has been showing very promising results in the clinic. And indeed, we can see that Xenostat is very effective at inhibiting tumor growth and maintenance in models from various cancer types. And it does this indeed by inhibiting cancer cell self-renewal and pushing cells towards differentiation. Now, importantly, you could see that all this is indeed primarily mediated by H10, because if we prevent re-expression of the protein upon treatment, we can do this by expressing shRNAs within tumors. Then we can see that the anti-tumor effect of Xenostat is completely abrogated. And personally, I found this result actually quite remarkable because This is one of the most pleiotropic drugs that one can think of. So it uses massive hypercirculation of chromatin across the whole genome. There are thousands of genes which are affected by treatment. But if we look for genes which are really important, truly important for mediating the functional consequences of this drug on cancer cells, well, this seems to come down to only one protein, and this is H10. Now, one thing that is quite exciting about this drug is that while it effectively inhibits cancer cell self-renewal, it does not interfere with normal tissue homeostasis. So here we're looking at 3 different tissues characterized by high cellular turnover, and in all of them, we couldn't see any difference in treated animals. And this is actually quite so very much in line with what's been seen in the clinic, where the drug has been shown to have a very sort of good safety profile. Now, the reason why normal tissues are unaffected by quisinostat is that actually normal tissue stem cells do express already H10. So here we're visualizing stem cells through LRG5 marker stem cell, both in the hair follicle and in the colon. And we can see that the cells express H10 as the more differentiated cells within the tissue. So this clearly shows that evidently H10 is not detrimental for normal self-renewing cells, and also explains why the cells don't really care about the drug. And this selective effect for cancer cells obviously is very important. This is something that has been actually very difficult to achieve when focusing, for instance, on developmental signaling pathways, such as Wnt or Hedgehog, because normal cells are equally reliant on these pathways to sustain the self-renewal. As although we do have some Q2 inhibitors, the problem is that they are quite toxic. But in this case, by focusing on the epigenetic state of cells, somehow we managed to find this selectivity. But to push this one step further, we also asked whether we could try to take advantage of the quisinostat H10 axis to inhibit disease recurrence, essentially trapping cells in a non-dividing state. So to do this, we use a number of models of acquired resistance to targeted therapy. So these are, for instance, lung cancer cells, which are so EGFR mutated lung cancer, that when treated with EGFR inhibitors in vitro, they very nicely mimic the response that we typically see in patients. So we see that the targeted agent is very effective at killing most of the cells within the population, but it's always most, never all. There are always a few cells that remain in the plate, and they sit there in a kind of quiescent state. But as soon as we remove the drug from their state, they start to divide again and reconstitute an expanding population. Now, if at this stage here we treat cells with quisinostat, however, we see that they fail to re-enter the cell cycle and remain in a non-dividing state. And importantly, this is even more effective than just keeping the targeted agent on cells, because at some point full resistance emerges and cells start to divide even in the presence of the drug. So this is lung cancer. Now, this is a completely different system. This is breast cancer that develops resistance to HER2 antibodies. The result is the same. Again, we prevent re-expansion of these few resistant cells. And we've also validated this in vivo. So there's now mice that develop spontaneous lung cancer, again, driven by EGFR. And we can track tumors, in this case, by CT scans. And again, when we give the targeted therapy, it's very effective in using tumor regression. Tumors almost seem to disappear. But pretty much every single case, this is just a transient response, and then tumors very quickly come back. However, if we treat these tumors, then with quisinostat, we see that most of them remain in a sort of undetectable, and the few that come back, they grow very slowly. So this result suggests that sequential administration of targeted therapy that kills most of the cells within tumors, and then quisinostat, which prevents the expansion of the few resistant cells, could be a valuable strategy to achieve prolonged response in patients. What is attractive about the strategy is that it's completely independent of the resistant mechanism, because we're not targeting the resistance per se, but actually the next step, which is the reconstitution of the tumor. And this is a universal mechanism of disease relapse. So at least in theory, this should be quite broadly applicable. So to summarize this first part, show you that we started from a very basic question. So what regulate cancer cell self-renewal, and why is it that not all cells are able to do that? This led us to identify a molecular mechanism that is relevant for numerous cancer types and relies on an integral component of chromatin. We also found a way to interfere pharmacologically with this mechanism, and also found that we can use a well-tolerated drug to inhibit disease maintenance and relapse without interfering with normal tissue homeostasis. Okay, so I'm going to switch now to the second topic and tell you a bit about our efforts in trying to understand why a disrupted epigenetic regulatory network is selected in cancer. So what you see here is the result of an unbiased analysis that's been performed a few years ago. And this looked for genes which are frequently mutated either in specific cancer types or in pan-cancer cohorts. And it's very striking how many of these genes are involved in the regulation of chromatin and DNA methylation. And interesting, in most cases, these are loss of function mutations, so the protein is gone, which is indicated here by the blue color. Now, some of these mutations are clonal, so they're present in the whole tumor, and this typically are selected early on in the disease. But others are subclonal, which means they occur later within established tumors. And these are thought to promote cancer evolution. There is a detailed view of two patients from two different cancer types, and these are phylogenetic trees. So we have here the clonal mutations within the trunk, and then there are these branches which represent the emerging subclones. And again, we can see how many of those contain mutations in chromatin regulators. Now, this is a more comprehensive analysis that we've done on 100 lung cancer patients that were profiled by the TRACERx study. And here we can see, again, subclonal mutations are really prevalent across all patients, and also that they spread across the whole network. So there is a class or a specific gene which are particularly affected. Now, the fact that we see these subclonal mutations means that they are in subpopulations that have expanded within tumors, otherwise we will not be able to see them. And so this suggests that this alteration may confer a competitive advantage to the affected cells. And so the question is, what is the favorable trait that is gained by epigenetically disrupted cells? Now, if we think about competition in established tumors, well, we need to keep in mind that cancer cells do not grow in isolation, obviously, but they are in a complex microenvironment that is characterized by distinct chemical, physical, and cellular components. And some of these features support the growth of cancer cells, but ultimately, this is really quite hostile and nasty environment, which is very crowded, and there's limited availability of nutrients and oxygen. And so all this makes it actually quite hard for cells to even survive and proliferate. So this exerts a strong selective pressure on the cell population. Another key feature of this microenvironment is that it's dynamic, so it co-evolves with the cancer, as the tumor cells need to constantly adjust to changing conditions. Now, considering the key role that epigenetic mechanisms play in physiology and mediating the response to environmental cues, we wonder whether the observed subclonal mutations may impact the interaction of cancer cells with the tumor microenvironment. As an experimental model, what happens within tumors, we initially focus on something that pretty much all cancer cells need to cope with, which is the heterogeneous and overall limited availability of nutrient in the crowded and poorly vascularized tumors. And to mimic that, we took cells from two cancer types, grew them under condition of nutrient deprivation, but it is by depleting either glutamine or glucose. And when we do this, we can see that cells don't really like it, they straggle quite soon. Most of them die eventually, but there are a few cells that somehow manage to survive and even proliferate in these harsh conditions, and at the end, they take over the population. So in this setting, we model on a large scale the loss of function mutations that we see often in patients. And to do this, we took advantage of a CRISPR library that were previously generated and targets over 300 epigenetic regulators and belonging to all the functional classes. And we have this array, this library in an array format. And so this allows us to basically systematically inactivate network components and then use microscopy-based readouts to see the effect of this inactivation on cell function. So using this experimental setup, we basically look for knockout populations that exhibit enhanced fitness when grown under stress. In this case, nutrient deprivation. And what we found is that a surprisingly large number of knockout populations did better than control cells in the setting. And control cells here are cells where we targeted known as stress genes. So here, these are a few examples that you see. First, this is the summary of the full data set, but green indicates enhanced fitness. And we can see that this is really a stress-specific effect. So we examine cells grown in unperturbed conditions. There's not much happening. But again, this phenotype basically emerges when cells grow under stress. Importantly, this effect is shared across all layers of epigenetic regulation. And so we can see here, basically, we're comparing the distribution of the targeted genes with the knockout populations that showed enhanced fitness. And we can see that the stress-specific phenotype is essentially shared. Again, it's not restricted to any particular class. So what we have here is basically it's a broad effect that is not restricted to any particular molecular function. And we can see that also is independent on the type of stress. So here we've done basically the same analysis, but now growing cells in acidic environments, which is another thing that cancer cells often experience within tumors, because it's a downstream consequence of hypoxia. And also in this case, almost 100 knockout population exhibited enhanced fitness, again, across all classes. Importantly, most of these knockout populations were resistant to both types of stresses, which have opposite effects on cells. And so this really indicates that it's not about a particular pathway that is specifically sensitive to epigenetic deregulation, but it's more about acquiring a phenotype that allows cells to more easily tolerate hostile environments. And indeed, when we look at these populations, we can see that there are more resistant cells in both conditions that manage to essentially survive and keep growing. And so something that happens sporadically in control cells now happens at a high frequency in epigenetically compromised cells. Now, this is all in vitro. What about a real tumor? And so to assess this, we selected EZH2 as a representative epigenetic regulator and performed an in vivo competition assay. Now, one of the reasons why we chose EZH2 is that it's a quite interesting case in cancer, because on one hand, we have a number of dependencies on these proteins have been reported in some contexts, which suggests a tumor promoting role. But at the same time, there are recurrent loss of function mutations that have been detected, which indicates that instead its loss may be beneficial for cancer cells. So here we see the experimental setup that we use. So we prepare a 1-to-1 mix of control and knockout cells, and we inject them in mice to induce tumor formation. As I mentioned earlier, nutrients are often limiting within tumors, but we wanted to maximize this. And so we treated tumors, so we waited until tumors appear, and then we treated them with an anti-angiogenic drug, which basically enhances nutrient deprivation and hypoxia by restricting vessel formation. So we grew tumors for about a month under treatment, and then at the end, we quantified the relative proportion of the two subset of cells. And as you can see here, the result was very clear. In all treated tumor, EZH2 strongly outcompeted the control cells. So at this point, we wanted to understand a bit more about the cellular mechanisms that lead to this stress-resistant phenotype. And so we tested a number of possible models. So one possibility is that inactivation of chromatin and DNA modifiers may promote genomic instability and generate genetically distinct subclones with increased resistance to stress that could be then selected. So if that's the case, one expectation is that if we restore proper epigenetic control after an initial perturbation, this should not affect the resistant phenotype because this is hardwired in the genome. So to test this possibility, we switched from genetic alterations to epigenetic inhibitors because we wanted to do this reversibly. And so we performed a washout experiment. This schematized here. So what we did, basically, we treated cells with a panel of epigenetic inhibitors, assessed the response in nutrient starvation after 9 days, then wash out the drug and examine their fitness after an additional 9 days growth, this time in the absence of the compound. And as you can see here, in all cases, we observed a reversal of the stress-resistant phenotype upon withdrawal of the inhibitors, which clearly demonstrates that the observed resistance is not genetically encoded. So the first model, genetic diversity, can be rejected. A second possible scenario is that a more permissive epigenetic landscape in knockout population may enhance phenotypic plasticity by favoring transition between cellular states. And so here, prediction is that knockout cells should be able to reversibly adjust their behavior in response to changing environment. And this is exactly what we see, both when we switch between unperturbed and stress condition back and forth, and also when we expose cells to different types of stress sequentially. So these patterns are indeed consistent with enhanced plasticity, but we really wanted to see this transition in sort of in real time and at a single cell level. So to do this, we took advantage of the fact that one of the consequences of nutrient deprivation is that cells downregulate oxidative phosphorylation, which is the respiration that occurs in the mitochondria and is the main source of cellular energy. And we were lucky enough that our colleagues in the high lab at the Crick just developed a FRET biosensor that allows monitoring of oxidative phosphorylation in living cells. Now, because the way the biosensor is made, the FRET signal is actually inversely correlated to the oxidative phosphorylation level. So we start with low levels here in happy cells before starvation. But as soon as we deprive nutrients, we can see that the signal goes up, which means that oxidative phosphorylation goes down. And this increases progressively up to 24 hours when cells basically reach a fully stressed state. So this is what we see in control cells. Now we repeated the same experiment with epigenetically compromised cells. And here again, we selected EZH2, this time mainly because the availability of specific inhibitors made it easier to do the experiment. So what we expected to see here was some kind of up and down in this line, which would suggest or would indicate fast transition between different states. But instead, what we got was a rather flat line, which indicates that the cells were actually quite static, so they remained much more similar to the initial state, and essentially did not reach a fully stressed state. So what we see here is now a decreased actually ability of transition between different states, which argues against the plasticity model, and instead suggests an alternative model whereby epigenetically disrupted cells are unable to efficiently respond to environmental changes. And since they fail to reach a fully stressed state, they're now more tolerant to unfavorable conditions. And interestingly, although this model is conceptually antithetic to the plasticity model, it could explain the results that I showed earlier equally well, because the cells are essentially oblivious to their surroundings and they don't respond to changes. They essentially can keep going regardless of the environment that they are in. So to gather evidence in support of this phenotypic inertia model, we perform a single-cell RNA-seq analysis comparing control cells and 5 knockout populations that displayed varying degrees of enhanced fitness. And the first thing that we did with this data set was to characterize the transcriptional response to stress in control cells. You can see in this heat map that's already at 1 day after nutrient deprivation, a lot of things have happened and cells have changed a lot. We detected about 3,000 genes that respond to stress. And this belonged to 2 main categories. So we have fitness signatures, which basically support cell growth and metabolism. We have, for example, MYC target genes, cycle genes, oxidative phosphorylation. And these undergo a coordinated and strong downregulation. And then we have stress genes, stress signatures, as expected, And this include NF-kappa-B or P53 targets, but also genes driving the apoptotic program, and these are upregulated. Now, a key aspect of all this is that we can detect these changes very early on, before basically defects in proliferation and survival are evident. So these changes are really causal in inducing the phenotypic changes. Now, if we look at these genes in the knockout populations, what we see is that the changes are there, but they're much milder, which indicates the cells remain more similar to the initial and unstressed state, supporting the phenotypic inertia model. Now, what's interesting here is that we're looking at epigenetic regulators with very diverse function. So we have member of the repressive complex, BRC2, then an integral component of chromatin. This is a chromatin remodeler, which is involved in transcriptional activation. So very different functions, but now we reduce their loss, and this surprisingly leads to very similar transcriptional alterations in this context. And I remind you that we've seen the same enhanced fitness in about 100 different knockout populations. And this matches what we see in patients, mutations being again spread across all functional classes. So the obvious question that comes to mind here is, well, what's the common molecular basis underpinning all this? And how come all those different alterations converge into a similar phenotype? Now, since we're dealing with such a diverse group of proteins, well, it seems unlikely that this comes down to the misregulation of a few specific genes. So we decided to examine something more general, so more general features of transcriptions that could be broadly affected by disruption of epigenetic control. As you probably know, gene transcription is discontinuous and occurs in pulses of activity, which are separated by relatively long intervals of inactivity. And this results in production of RNA molecule in discrete bursts, whose frequency can vary from just a few minutes to several hours. And this is due to both the randomness of intermolecular interactions, which is required to initiate transcription, but also to packaging of DNA into chromatin, which regulates accessibility of regulatory regions. Now, broadly speaking, the size of this burst, so how much RNA is made at every round, is dependent on promoter architecture. So, for example, how many transcription factor binding sites are present, whereas frequency is instead more related to enhancer activity. Now, single-cell RNA-seq data sets quite conveniently can be used to estimate those kinetic parameters on a genome scale. And so this basically gives a kind of a snapshot of transcriptional activity. So if we do this in unperturbed conditions, we get expected patterns. So we can see, for example, highly expressed genes as expected to have both high frequency and high size. But if we look at it at the global level, we can see that the relationship between all these parameters is not a simple one. So there's a lot of structure in this data set, and there are clearly some genes which rely more on frequency and other more on size to regulate their expression. So the first thing that we asked with all this was, well, what happens to these parameters when control cells, so which have an intact epigenetic regulatory network, experience stressful conditions? And the answer is that there is a striking reduction in burst frequency, which means that global transcriptional activity is strongly inhibited. And again, this happens early on before phenotypic changes can be detected. Now, this is a global effect, but actually genes are not all affected in the same way. And those that change the most are those characterized by high frequency, which makes sense if you think about it, because obviously a gene that fires every 5 minutes will be more affected by blocking transcription than one that fires every 4 hours. But the interesting thing here is that among these high frequency genes, we find all the fitness genes that are affected upon stress. So they make target genes, oxidative phosphorylation, cell cycle genes. They're all here, the sort of high end of the distribution. They're all very sensitive to this global transcription inhibition. And this nicely, I think, explains their coordinated downregulation upon stress. Now, if we look at burst size, there's not much happening globally, but stress genes are actually quite different, and they show a progressive increase, which is likely due to the binding of stress-activated transcription factor to their promoters. And interestingly, these stress genes are actually low frequency genes, so they are on this side of the graph, and so they're not particularly affected by the global reduction in frequency. So basically, a normal response to stress involves a global reduction in burst frequency, which affects primarily high-frequency fitness genes, and a specific increase in burst size at stress genes. Now, what happens in epigenetically disrupted cells? Well, not much. So we can see that fitness genes continue to be transcribed at high frequency, and stress genes are unaltered early on and only show mild changes at later stages. And we can see these patterns really across all the different knockout populations that we had examined. But this is not just modeling. So, for example, if we measure nascent RNA levels, we can see the fitness genes stop being transcribed upon stress. And when we do chip of RNA polymerase, we see that this is lost from gene bodies in control cells, but is fully retained in the knockout population. So to summarize this into a model, we've seen here that when cells experience stressful conditions, they mount a complex response that in the early stages essentially aims at saving energy and biomass, and they do this by halting proliferation and decreasing metabolism. And if this works and they find a way to overcome the challenge, well, good, they survive. Otherwise, they commit to apoptosis. And at the population level, basically, this is a pro-survival strategy that aims at getting rid of unfit cells. At the transcriptional level, this response is achieved through a global reduction. Transcriptional activity mainly affects genes which are transcribed at high frequency, which includes fitness genes, and activation of stress pathways by a modulation of burst size. And various components of the epigenetic networks are involved in this process and are used to execute this transcriptional rewiring. However, when the network is defective, regardless of where the problem is, these transcriptional changes cannot happen as efficiently as they should, and cells fail to alter their phenotype in response to stress. And so what we refer to as transcriptional numbness of epigenetically disrupted cells leads to phenotypic inertia, and this in turn increases their tolerance to environmental stress. So the obvious analogy that comes to mind here is with p53 null cells, which are insensitive to cell intrinsic stress, and even if they are full of DNA damage, they just don't realize and they keep going. And something quite similar, we think, happens also in this case. And what is fascinating to me is that in both cases, loss of functions basically turn into a fitness advantage by cancer cells, which essentially don't care about things, doing things in the right way. So I'm going to finish here with a thought. So I'll show you two examples where loss of function of epigenetic regulators is beneficial for cancer cells. On one hand, to sustain self-renewal, and on the other one, to cope with environmental stress. At the same time, we know that cancer cells can develop dependencies on some of these proteins, and when we inhibit their function, well, cells don't like it. And so how do we reconcile all this? Well, I think one important aspect is context here, and I think EZH2 illustrates this perfectly. So we know the drug has a lot of mutations in multiple PRC2 members in patients, which means that their loss is not only tolerated, but it's actually beneficial in many cases. Now, when it comes to EZH2 inhibitors, sensitivity certainly not universal. It's actually quite difficult to predict when a patient or a cell line will respond to treatment. So it's likely a combination of genetic background, tissue of origin, and also disease stage would really create dependencies on EZH2. And the second point is that epigenetics integrate inputs from both cell intrinsic and cell extrinsic sources. And the balance between these two really determines the outcome of an interference with its mechanisms. And as a general statement, I think this is actually really the challenge with epigenetic therapy. So the fact that there are multiple forces at play, sometimes they even oppose each other, and it's not always easy to control them all. And so with this, and just quick acknowledge that the people who've done the work. So for the H10 part, Cristina Morales is the person who has mainly contributed to the study. She's a senior scientist in the lab. Whereas Yanis Lucas is a PhD student, worked on the study environmental stress, also with help from other lab members. And stop here. Thank you all for your attention.
Thanks, Paola. We already ran out of time, but maybe we can still, what do you think, Santi? We can take still a couple of questions, no? Yeah. Would you like to read the first one, Santi? We cannot hear you.
No, but we can start, Luciano, no problem.
Okay. So you want to read the question, or you want to, I can read it.
Yeah, I can read it. Tony Sork asks you, Paola, is it known how quinostat upregulates H1? Is it direct effect through increasing histone acetylation at H1 gene loci?
Yes, that's the case. So basically, it's so we've seen that acetylation of core histone is required for upregulation of H1 This happens at the transcriptional level. So, yes. Yeah,
I actually have a question related to that. So what you presented is that in several cancer model systems, there is downregulation of histone H1. So how this is achieved? So how the oncogene leads to repress the expression specifically of histone H1 and all other HNS 1.0 and all others variant?
Yeah, so mechanistically, we don't know. So what we've seen is basically our observation is that diverse, so many different oncogenes basically correlates with this downregulation. What we think is happening is that it's a selection process. So basically only cells, because it's so strong basically in blocking proliferative potential, only the cells that maybe stochastically manage to downregulate this then are selected during tumor growth.
Okay.
Yeah, Omne Rod asks you, first of all, thank you for the nice, interesting talk. Can you comment on the phenotypic inertia? And what is your opinion about why normal cells will respond so strongly to environmental stress, where the number cells are capable of functioning in that environment? What is your comment on that?
Sorry, it was what? The normal cells, why do they respond so strongly?
So strongly, yes.
Yeah. Well, that's basically, it's an evolutionary, selected response to stress. So cells are programmed to actually respond very quickly and very strongly to stress. Again, if you think in a normal population, this is, again, it's a way for the population to get rid of unfit cells. And so if you manage to respond and cope with the stress, then you will keep going. Otherwise, the population, prefers to get rid of the cells. So this is something that we see in nature at all levels. And again, the point is that cancer cells don't do this in the right way, and they're still fine with it.
Yeah, I think now due to the limitation of time, we should move on. What do you think? And then I can forward you later on, Paola, the questions. There are quite a few questions that would be great.
Yeah, sorry, at the beginning I had this technical issue.
No worries.
Okay, thank you.
Thanks, Paola.
Thank you very much, Paola. Thank you. It was great.
So